Dear students, in this section, I am going to discuss about the carbohydrates. This is the main energy source of our nutrition. The carbohydrates are the most abundant organic constituents of plants. Basically, the carbohydrates are synthesized in the plants during photosynthesis, and they serve as the major source of chemical energy for living organisms, as well as the important constituents of the supporting tissues. This can be acts as the building block of sub tissue. The carbohydrates consisting of the molecular formula CXH2O whole Y, where the X and Y may be equal, may not be equal, and they are usually defined as the polyhydroxide aldehyde and ketone on the depending on the presence of the uh, functional group, whether it is aldehyde or ketone, or the substance that hydrolyzed to yield polyhydroxyaldehyde and ketone. And these simple carbohydrates are known as sugars or saccharides, and the ending of the name of the most sugar is with O's. For example, glucose, fructose, sucrose, maltose, these are the all carbohydrates. Among them, glucose and fructose are monosaccharides, whereas sucrose and maltose are the disaccharides. So the classification is the simple carbohydrates containing monosaccharide and disaccharides, and the complex carbohydrates are oligosaccharide and polysaccharide. The monosaccharides are consisting of only one unit of or one unit of monosaccharide, and disaccharides consisting of two monosaccharide units, and in the complex carbohydrates like oligosaccharide and polysaccharide, more than two monosaccharides are there, and these monosaccharides are joined together by the covalent bond known as the glycosidic bond. Upon the hydrolysis, these disaccharide and polysaccharides are broken down into two or multiple monosaccharide units with the elimination of hydrogen, with the elimination of water. And the monosaccharides consisting of only one saccharide molecule, that are non-hydrolyzable further, and these are further classified according to the number of carbon atom present. If there is carbon atom is 6, then it is called as hexose, but when the carbon atom is present is 5, this is known as pentose. For 4, it is tetrose, and so on. And the carbonyl group containing the type of carbonyl group contained on the basis of this containing carbonyl group, we can further classify this carbohydrate in two categories known as aldose and ketose. In this section slide, I have described the molecular structure of carbohydrate monosaccharides, where I clearly depicts the long chain monosaccharide can undergoes the ring structure, and the presence of aldehyde or keto group in the monosaccharide unit gives the name aldose and ketose, and also the number of carbon atoms gives the name, that is here the 4 carbon atom present, so this is tetrose, and also the aldehyde group is here, so it is known as aldotetrose. Such as and similarly, this is aldotriose, it is ketotetrose and ketopentose, because there is keto group present, and the number of carbon here is 5, and it is 4. So this is the slide where I have described the monosaccharide classification basically. And the monosaccharides are used in the pharmaceutical industry, and so it has very much importance in this industry to produce the medicine and injection and the nutrient. And also they are used for the sweetening agent it is also used in the ice cream and candy industry, as well as in confectionery industry. The disaccharides are the carbohydrates that yield two monosaccharide molecules upon hydrolysis. That's a sucrose, lactose, maltose. This is the sucrose molecule shown here, where upon hydrolysis we can get the glucose and fructose molecules, and these glucose and fructose molecules are joined together by the glycosidic bonds, and this is the alpha 1,4 glycosidic bond. Because this is one number carbon is joined at the fourth number carbon here, and this is known as 1 2 3 4 number carbon. This is known as the alpha 1 4 linkage, alpha 1 4 glycosidic bond. And these are known as these disaccharides known as the glycosides. So as they are consisting of glycosidic bonds, so these are known as glycosides. The polysaccharides are also known as glycans because they are consisting of monosaccharides, multiple monosaccharides joined together by the glycosidic linkage. The polysaccharide that are polymer of a single monosaccharide are called homopolysaccharide, and those are made up of more than one type of monosaccharide are known as the heteropolysaccharides. The homopolysaccharides are also classified on the basis of their monosaccharide units. A homopolysaccharide consisting of glucose monomeric unit is called the glucan, or one consisting of galactose unit is known as the galactan, and so on. Basically, there are three main categories of polysaccharide present in the nature. These are cellulose, starch, and glycogen. In case of animal, the glycogen is found, and the rest of them are found. Rest of the polysaccharides are found in plant system. We can see that amylopectin and glycogen are branched here, and they are consisting of alpha 1 4 and alpha 1 6 glycosidic bond. And in case of no unbranched polysaccharide chain, only 1 4 glycosidic bond is present. Uh, Now, I previously told that carbohydrates are used as a building blocks. In case of plants, the starch is used as a building block of plant seeds. Where it is very much common in plant seed, and the glycogen, which is present in the animal system, they are main building unit of the muscles, with the formation of some muscular, with the addition of the muscular protein, the glycogen forms the muscle. And in case of bacteria and fungus cell wall, we can found the dextran, which is a polysaccharide. It is made up of alpha 1 4 1 6 poly D glucose, and have 1 3 bunch, and they also have some alpha 1 2 and 1 4 branches. In dental plaque formed by bacteria growing teeth, here the dextran deposition can be observed, and sometimes we can use the synthetic dextran in the column chromatography. The column is made up of synthetic dextran known as Cefadex. This is the dextran molecule. The N represent with the number of this type of molecules. So this is a polymer. So N can be multiple times we use this N, and this long chain is mainly basically the structural unit of the bacterial and yeast cell wall. The cellulose is present in the plant body. It is basically present in the plant cell wall. It contains the beta 1 4 linkage, glycosidic linkage, and the chitin mostly found in the crustacean exoskeleton, like shrimp, crab. This type of animals are known as the crustacean, and their exoskeleton is made up of the chitin, which is joined by the this type of linkage, and this is a polymer of glucose, amylopectin basically, and this is homopolymers of acetyl glucosamine, and this is the acetyl glucosamine, and when they are linkage in between them, they form the polymer of chitin. The bacterial cell wall also consisting of the two very special type of carbohydrates, polysaccharide. They are known as N-acetyl muramic acid and N-acetyl beta D glucosamine. These two molecules, these two polysaccharide present in the these two monosaccharide present in the bacterial cell wall to form the polysaccharide unit, and in an alternative way, and by presence of this type of molecule, that we can classify the bacteria in two classes, like gram positive and gram negative bacteria. While in a gram positive bacteria, the pentaglycine chain is the cross link between which is present. This is the pentaglycine glycine chain, which is absent in case of gram negative bacteria. So students, this is the class where I have discussed about the carbohydrates. So please go through this slide. If you have any question, you can ask me later. So thank you, students.